So we just finished talking about the brain stem, which has the medulla oblongata, the pons, and the midbrain. Now we're going to focus on the diencephalon, the cerebrum, and the cerebellum. First, let's dive in and talk about the cerebellum. When we look at the cerebellum, the root is associated with the cerebrum. And not only does it just look like a smaller portion of the cerebrum, it also is going to have little sulci, little gyri, little fissures. In many respects, it just resembles a little cerebrum or a little brain. And as we look at the cerebellum, it's going to be located on the posterior of the brain stem. And as we zoom in on it, our cerebellum has a very high concentration of gray matter. There is almost no white matter within the human cerebellum. The human cerebellum is overwhelmingly gray matter, and this contributes to the very high density of neurons. Our cerebellum has approximately half of the neurons of our body packed into this little tiny region. As we look at our cerebellum, it has a left and right hemisphere associated with it, with a small bridge in between known as the vermis. And as we look at it in more detail, we have a lot of branching white matter within the cerebellum. This branching white matter was thought to resemble a tree, the arborvitae. You may remember that term from lab. Within our cerebellum, we have peduncles, just like we had the cerebral peduncles of the midbrain connecting the midbrain to the diencephalon and the cerebrum. Within the cerebellum, we have peduncles connecting the cerebellum to the brainstem. These peduncles are superior, medial, or middle, and inferior. The inferior peduncle is going to be the connection point that's down low. Superior will be the top connection point, and then the middle peduncle is going to be the lateral connection point. As we look at these peduncles, these are typically going to be white matter that relay information to and from the cerebellum. Our inferior peduncle is going to link to the inferior portion of the brainstem. It'll link to the medulla oblongata. The middle peduncle will link to the pons, and then the superior peduncle is going to link to the superior part of the brainstem. It'll link to the midbrain. As we look at these peduncles, the inferior peduncle that's closer to the spinal cord will be linked or associated with spinal cord input. The middle peduncle, which is associated with the pons, is going to have a mixture of input from the rest of the brain and a little bit from the spinal cord. So because it's in the middle, we're not going to have specialized function with either the cerebrum or the spinal cord. But when we look at the superior peduncles that head up to the cerebrum, they are going to focus primarily on cerebellar output or connections to the higher brain functions. When we think of the cerebellum, I want to emphasize to you motor coordination. More than anything else, our cerebellum allows for us to have motor coordination. There's a condition associated with chronic alcoholism. There's certain populations of people that have just drank way too much alcohol, they've lost way too many neurons, and consequently, they have constant tremors associated with the loss of neurons, not only from their basal nuclei, but significant degradation of their cerebellum as well. So if you're ever talking to an individual and they're constantly twitching, it may be Parkinson's disease, or it may just be chronic alcoholism that has caused degradation of these motor centers in their brain. We also use our cerebrum for other things as well, our cerebellum for other things as well. We use it to compare textures of objects. We use our cerebellum to focus on linguistic control. When we think of linguistic control in particular, there's a lot of motor movements associated with speech and communication. Think of your tongue, your lips, your eyes. There's a lot of regulation of that motor control. One of the things that I like to emphasize with the cerebellum is that we have motor memory. You can train yourself to have automatic skeletal muscle movements. And as you go through the practice, you can think of athletes drilling constantly, or drilling to move, or practicing to move over and over and over again. Or a lot of you have motor memory. When you sit down in your car, you keep turn the ignition, reach down for the shift, lot of you have just been trained to reach for the shifter. And then when you sit in a different car that maybe doesn't have a shifter down low, it has a shifter up on the steering column, you're reaching down there for the shifter and there's nothing for you to grab. Or if you go from driving stick shift to driving a manual automatic transmission, you have the muscle memory of pressing the clutch whenever you want to shift gears, but it's not there, and it takes a while to overcome that muscle memory. We also will use our cerebellum to judge the differences between tones and distinguishing between pitch. Individuals that have a strong musical talent, or if you have the ability to read inflection, you have a more developed cerebellum than individuals that are potentially tone deaf or potentially have a hard time reading those nonverbal social cues. You know, the tone, what's the cliche? It's not what you say, it's how you say it that's important. And the reason for that, picking up on those differences in tones, can be linked to the cerebellum's function. It's not to say that if you don't have it, you'll never have it. It just means that you have to train and you have to practice and teach your cerebellum these abilities. We also find that our cerebellum is going to be linked to hyperactivity. Individuals that have been diagnosed with ADHD, preferentially or by proportion, will have a smaller individuals that have not been diagnosed with ADHD. So let's review our big structures of the brain again. We have the brain stem, we have the diencephalon, we have the cerebellum, and then we have the cerebrum. And as we look at the diencephalon, that's the one that there's the most debate about where to categorize. As we're looking at the diencephalon, it arises from a structure during embryonic development known as the forebrain. And within our forebrain, we'll have the cerebrum or telencephalon, and then the diencephalon within the central portion of that. The biggest structure of our diencephalon is the thalamus. It makes the largest volume and has the largest mass of the diencephalon. But there are other parts of the diencephalon as well. We also have a hypothalamus and an epithalamus as well. Now, when we look at the thalamus, it has a bunch of nuclei within it, a bunch of localized regions that have concentrations of neuronal cell bodies within the central nervous system. And these nuclei within the thalamus all serve specific functions. At a gross level, when we think of the thalamus, I want you to think of it as a relay station. The thalamus is going to receive a lot of information from the superior peduncle of the cerebellum. It also will receive a lot of information from the cerebral peduncles of the midbrain. And then information is sent up to the thalamus, and then the thalamus, which is part of our reticular network, is going to process that information and then send it to the appropriate part of the brain for the final decision-making process. If we back up a slide and look at the gross anatomy, let's shift colors here. Let's go with red. Predominantly, information is sent to the thalamus of the diencephalon, and from that thalamus, the information is then relayed to the rest of our higher brain functions. When we look at our thalamus, we have a left and right thalamus that correspond to the lateralization of our higher brain functions. When we think of our forebrain, there's the left and right hemispheres of the cerebrum, and we have the left and right sides of the thalamus of the diencephalon as well. When we look at our thalamus, I can't emphasize this enough, it filters information. It's the gatekeeper. It determines what goes to our cerebrum and what leaves our cerebrum in most situations. So as we're looking at the thalamus, it will process that information and control what goes in and what goes out. Our thalamus is also going to play a key role of the reticular formation. And as it's part of that reticular formation, it will relay information from the higher brain to the lower brain and down to the spinal cord. We also are going to have within the thalamus some feedback loops that occur between the cerebral cortex. In particular, we have the motor centers of the cerebral cortex and some filtering nuclei, the basal nuclei, that help to process out the unwanted motor signals. When the basal nuclei are functioning appropriately, we have smooth control over our body, we have good tone, we have good posture. As we lose the basal nuclei, we start to develop those twitches and tremors. Something else that our thalamus is intimately involved with is the limbic system. The limbic system is not so much a structural categorization, but is primarily a functional categorization when we think of the limbic system. When we think of the limbic system, I want you to think of the emotional brain. Strong emotions, strong desires, anger, libido, hunger, satiation. These are all associated with the limbic system. And the limbic system encompasses many different structures through many different parts of the brain. It's it's a functional categorization within the brain. Within our Diencephalon, we also have the hypothalamus. We've talked about the hypothalamus already with our endocrine system discussion. The hypothalamus releases a lot of hormones that travel to the anterior pituitary gland and stimulate the anterior pituitary to make new hormones, or those hormones will travel to the posterior pituitary to be stored in the posterior pituitary. As we look at our thalamus, it is located right here within the Diencephalon. Connecting the thalamus to the pituitary gland, we have a small band of tissue known as the infundibulum. And then on the inferior margin of the thalamus, there are two spherical structures that early anatomists thought resembled breasts. Those structures are referred to as the mammillary bodies. And on the inferior anterior portion of our thalamus, we have the optic chiasma, or the optic chiasm. This is a region where the two optic nerves reach the brain. I mean hypothalamus. Thank you for calling me out on that one. I mean hypothalamus on this slide. So on the inferior anterior margin of the hypothalamus, we have the optic chiasma, and then the mammillary bodies over here for the inferior posterior margin of the hypothalamus. As we look at the autonomic nervous system, our hypothalamus is also going to play a major role in regulating the autonomic nervous system, particularly when we think of the homeostatic functions of the body. Our hypothalamus is one of the parts of the brain that can directly access the bloodstream. And because this part of the brain can directly access the bloodstream, it can monitor blood chemistry. We think of blood oxygen concentrations, blood pH, blood CO2 levels, glucose concentrations. These are all things that we regulate with our blood chemistry. And our hypothalamus, since it's part of the brain that can access the bloodstream, will be essential for maintaining that relatively constant internal environment in our bodies. So we mentioned the hormonal secretions, the hormonal functions we already emphasized on 2 slides ago. We also have some autonomic effects of our hypothalamus as well. One of the interesting autonomic effects of our hypothalamus is going to be the regulation of body temperature. The hypothalamus is one of the last regions of the brain that is going to be developed during gestation, and it's typically at about 32, 34, 36 weeks, depending on the individual, after fertilization, that the hypothalamus is able to regulate internal body temperatures. Individuals that have been born prematurely don't have this ability with their hypothalamus yet, and we consequently will see prematurely born infants put in incubators to help externally regulate their body temperatures, while their hypothalamus develops the ability. We're also going to regulate heart rate, blood pressure, the movement of secretions within our gastrointestinal tract, and just the movement of substances throughout our body. Part of that is going to be the vasoconstriction of the blood vessels or peristalsis within the digestive tract. So I'd already emphasized thermoregulation. Our hypothalamus is also going to regulate the formation of short-term memory and conversion to long-term memory. The hippocampus is a separate structure typically associated with the limbic system, and our hypothalamus will take the short-term memories of the hippocampus and help to convert those short-term memories into long-term memories. We're also going to use our hypothalamus to regulate our sense of thirst by regulating our blood chemistry. If our blood osmolarity becomes too high, if our salt concentration becomes too high in our blood, our hypothalamus is going to start to produce antidiuretic hormone, and that ADH will ultimately cause us to retain water and to feel a sense of thirst. We're also going to regulate our sense of hunger with our hypothalamus. As our hypothalamus is monitoring blood chemistry, it looks for nutrients in the bloodstream and will help dictate whether we feel hungry or we feel satiated. And then finally, we also are going to have some emotional or sexual responses associated with the hypothalamus. Our hypothalamus is part of the limbic system. It helps to regulate libido and sexual response in individuals. We also have the epithalamus as the diencephalon. The epithalamus, in most situations, is going to be used as a synonym with the pineal gland. The pineal gland being an endocrine gland that makes melatonin to regulate our circadian rhythms. But there's technically a secondary structure that almost no one mentions with the epithalamus, and that is the habenula. And our habenula serves as a relay system between the limbic system and the midbrain. This habenula is going to be located right here on the slide and serves as the superior roof of the third ventricle that circulates our cerebral spinal fluid from the lateral ventricles. And then finally, we have the cerebrum. Our cerebrum is the largest, most complicated part of the brain. And there's the most time dedicated to the cerebrum. To back up a little bit, the cerebrum is the part of the brain with the sulci and gyri. It has the wrinkles in it. It's what most people typically think of when they think of brains. And as we look at our cerebrum, it's our seat of perception. It's going to be where we have our sense of self. This is really what separates human beings from all of the other organisms on the planet. The relative size of our cerebrum in relationship to our body volume. Relative to our body volume, and in particular relative to our spinal cord volume, we have a very large cerebrum, larger than any other organism. And as we look at our cerebrum, it's going to have the left and right hemispheres separated by the longitudinal fissure, which you've covered in lab. And then there's a whole bunch of white matter with fibrous tracts in it. To review, in the central nervous system, when we have a tract is a bundle of axons in the central nervous system. In the peripheral nervous system, we call those nerves. And all of these tracts are grouped together to form a structure known as the corpus callosum, which connects the left cerebral hemisphere to the right cerebral hemisphere. The gyri and sulci of the cerebrum are much larger than the sulci and gyri of the cerebellum. And this is where we can squeeze in extra cell bodies of neurons, extra neurosoma. The function of the sulci and gyri are to increase surface area so we can increase processing power. As a rule of thumb, the wrinklier your brain is, the more sulci and gyri you have, the more intelligent you are, the more abilities you're going to have. Smooth brains are typically associated with mental retardation and or very low IQs. One of the really interesting things that we notice with the sulci and gyri is that as you learn new skills, the sulci and gyri will change shapes. There was a very well-published study that looked at brain scans of individuals before and after they learned to play the piano. And they all had the same new ridge form in their occipital lobe, the same new sulci and gyri. And then on the other hemisphere, the same study was done for individuals before and after they learned to play violin, and they found that the same new ridge formed in all of those individuals. Here are the lobes of the cerebrum, and there's a lot of information on the slide. We could camp for a long time on the slide. We're going to talk about the highlights, and then dive in and talk about functions in more detail on subsequent slides. On the anterior portion of the brain, anterior superior, we have the frontal lobe associated with the frontal bone. We have the parietal lobe associated with the parietal bone, and then occipital for the occipital bone, and then the temporal for the temporal bone. The only lobe of the cerebrum which is not named for a bone of the cranial vault, insula. To get to the insula, you need to pull back the temporal lobe and the frontal lobe to reveal another lobe of the brain underneath. As we're looking at these lobes of the brain, as neuroscience has advanced, we find that it's becoming increasingly complicated in that one lobe will have many, many different functions. We're going to talk about the few specific functions associated with each lobe, but while we're on this slide, let's talk about the central sulcus. The central sulcus emphasizes clarity and separates the frontal from the parietal lobes. And then the gyri directly anterior, central gyrus. Gyri directly posterior is the post-central gyrus. And we'll talk about those specific gyri in more detail. So when we think of the white matter of the cerebrum, our white matter of the cerebrum makes up a very large portion of it. These white matter portions of the cerebrum are heavily myelinated axons composed of tracts that are going to send information from one part of the brain to another part of the brain. And as we're looking at these tracts, or this way of moving information from one part of the brain to another part of the brain, we will send it different directions. These different directions are going to be carried on projection tracts, commissural tracts, and association tracts. When we look at a projection tract, that's going to be a tract that goes between our higher and lower brain functions. So these projection tracts are going to go from the surface of the cerebrum down typically to the spinal cord. So these are going to be tracts that are going to all head through the thalamus down to the spinal cord. When we look at association tracts, these are going to go from one part of the cerebral hemisphere to another part of our cerebral hemisphere. So to shift colors, I'll go with blue for the association tracts. We can take some sensory information and connect it to our motor information with an association tract. We could have a decision to comprehend language and a decision to talk about it, so that information from one part of the cerebrum to another part of the cerebral hemisphere. So we have projection tracts that connect the higher brain functions to the lower brain, with association that make connections from one part of the cerebral hemisphere to another part of the cerebral hemisphere. And then we have the commissural tracts that connect the two cerebral hemispheres to each other. And when we think of the commissural tracts, they connect a left brain to a right brain. They go back and forth between the two halves of the cerebrum. And as we look at those commissural tracts, they are overwhelmingly going to be located in the structure that connects the left and right hemispheres. The corpus callosum is overwhelmingly going to be made of the commissural tracts that connect the left and right hemispheres of the cerebrum. As we look at the cerebral cortex, we have gray matter, superficial on the cortex of our cerebrum. This gray matter is where we're going to have most of the processing occur, and within that gray matter, we'll have a high density of dendrites and a high density of neurosomas. We're also going to have, as we travel inferiorly to the tracts, with the commissural tracts and the association tracts. There we go. Yeah, I was getting some nonverbals from the classroom. Let's talk about neural integration. Slide. And I click through two. Here we go. Let's talk neural integration. So as we look at our neural integration, guys, gals, we're going to find that the gray matter is where we process information, where we integrate the information in our brains. We have most of our gray matter in our brains located at the superficial margin of the cerebrum. So the top of the wrinkly part of our brain is going to have most of our gray matter. We also have a very large amount of gray matter within the cerebellum as well. Our cerebellum is, by volume, mostly gray matter. And then deep, buried inside of the cerebrum, we have little regions of gray matter. And those little regions of gray matter are referred to as the basal nuclei collectively. Within the cerebral cortex, or the surface of the cerebrum that has the gray matter, this cerebral cortex is not very thick. So it's less than a centimeter thick, 2 to 3 millimeters thick. And it's going to have a lot of our gray matter for processing purposes. It doesn't have the 100 billion neurons of the cerebellum, but it still has a high amount of neurons. There's a lot of processing power associated with there. And we're also going to have the neocortex. This neocortex is the layering of our cortex that we typically as humans are going to have very well developed. Another way of thinking of this neocortex is to think of it as the new brain. When we think of the brain stem and the diencephalon, they're often referred to as the primitive brains or the reptilian brain. They carry out basic life functions that you need to stay alive. When we think of the neocortex, this is, from a developmental perspective, something that's relatively new that we as humans have developed, but most other species have not yet developed. And consequently, we have some extra functions. This will be our last slide for today. So as we think of this neocortex, or this highly developed cortex, cerebral cortex of the brain, we have the gray matter on the surface that's 2 to 3 millimeters thick, and then we're going to have multiple cell types, multiple kinds of neurons that integrate the cerebral cortex with the basal nuclei and the other regions of the brain. We have stellate cells, and our stellate cells are going to be typically associated with sensory input. As we look at the stellate cells on this figure, these stellate cells are multipolar neurons that are located in the fourth layer of the cerebral cortex. And then we'll also have pyramidal cells. These pyramidal cells serve to link the multiple layers of our neocortex. In lab, we had demo slides set up with the pyramidal cells that have long dendrites that extend up to the surface, and then have long axons that extend away to the white matter. And then they'll also have a few shorter dendrites that integrate with each other. When we think of these pyramidal cells, they get their namesake, pyramid, from the triangular shape of the neurosoma, and they serve to integrate the gray matter on the surface of our cerebral cortex with the gray matter of the basal nuclei buried deep within the cerebrum. And we're basically out of time for the day. We'll pick this up on Wednesday. We'll also spend some time on the in-class survey on Wednesday as well.